recording a little bit before time for the actual lecture to start. But I'm just going to remind you, as usual, to get the points for this video, I'm going to say a series of attendance words randomly throughout the video. When I do, you're going to write those down and take a screenshot of when I say those attendance words. Then, when I'm done with the lecture, email me with that list of attendance words. Email me, and also in the email include the screenshots. And then finally, the last thing I need to do is ask one question about the material covered in the lecture. So, the first word for attendance is going to be, well, it's not a word at all, but the letter N, as in November. N, I'm pointing at right there, is the first word for attendance. So write that down, take a screenshot, me pointing at the letter N. And now, since it is 7:45, you can go ahead and skip ahead the video about 10 minutes, and that's when I'll start the course announcements. And then about 5 minutes after that, we'll start the actual lecture. Morning.
So we're not doing the exam today?
No. Unfortunately, with the weather being the way it is, we, won't be able to. Or haven't had the time to get all the material covered.
Yeah.
You guys have any issues getting here with the weather on the roads, or is everything fine where you?
Yeah, everything's good. Cross lines.
Beard is here. Let's see. Ashley, right? Okay. Lucas?
Yep.
Okay. A little less than 5 minutes, I'll at least do the redundant announcements with the nothing new stuff. Like, for example, attendance is mandatory and participation is graded. If you're watching the video at a later date, just do what you usually do. Send an email every time I say an attendance word, write it down, take a screenshot of that moment, and then send me an email that has the screenshots and the list of words, in addition to one question about the material. If you're live streaming, which no one is right now, that I know of, but I'll say it just in case. You're going to do all that, except I don't need the screenshots. So do send the email before 9 a.m. So you don't need the screenshots. And of course, if you're here in person, today I just ask that you email me. Just a simple email with a date that says I'm here. Now, of course, if you want to, you can include all this other stuff to get a little bit more, a little extra credit with your attendance points. Yeah, so basically business as usual. See what that sound was before I move forward. Good, yeah. Anyway, so yeah, real quickly, again, if you're watching right now online, as opposed to watching a video at a later date, you're going to send me an email of all the attendance words that I say throughout the video, and then again, just one question about the lecture. Anyway, obviously check your emails and announcements before you come in. I'm sure you guys have. We've been doing it a lot recently. Yeah, nothing new there. So just a reminder, the biological molecules lab and the diffusion and the osmosis pre-lab are due today. On one hand, a lot of people are getting behind, right? They're getting behind on doing these assignments. So, I want to say, hey, be careful of that. Don't get too far behind. It's going to hurt you. On the other hand, I don't want you to feel bad about it because this has been a crazy semester. So don't feel bad if you're behind. Just know that you need to catch up, and then I'm here to help you. And I know it's really tough calling, changing things at the last minute thanks to weather and stuff like that. So don't feel bad about it. We're doing the best we can. I'll be flexible. And again, I'm here to help you. So far, nobody, I don't think, maybe one person, one or two people have asked for a little help, but no one said, hey, I don't know how to do this lab. Can you help me? So feel free. I prefer it. You're not bothering me. I don't think you're less of a student. It would be easier for me to walk you through a lab. That way, when I grade it, it's like, oh, that's full credit. Nice and easy. Rather than have to find what's wrong and leave feedback for what's wrong and then write a little thing about how you can resubmit with corrections. Matter of fact, since we're in person, from now on, I probably won't do that, just so know it's given. You can always resubmit with corrections. You can always resubmit with extra credit. But I'm going to quit writing that each time I grade one of your assignments. So the offer is still always there, but I'm just to save time, I'm not going to do that. So yeah, let me know if you need help with that. The big news, and this is actually news, and this is not old news. This is a new announcement, not a reminder. Obviously, we're not doing the exam today. We haven't covered the material that needs to be covered. So I'm pushing it back. It's open until Wednesday. We'll see. But just like we talked about it previously, when we get closer to the day of the exam, or the scheduled day of the exam, if any of you feel strongly about pushing it back even farther, let me know and I'll consider that. For example, if we finish this chapter on Monday, which I hope we don't, hope it's today. But if we don't finish it until Monday and you think that's not enough time to study for the exam on Wednesday, like I said, email me if you feel strongly enough about it. And enough people agree, then I might push it back a little bit, but we'll see. So any questions about any of this? All right, well, when we do have the exam, like I've been saying, when we have the exam, make sure you bring your computer. If you don't have one, let me know. Probably going to want to bring a charger because you don't want your computer to die. If you want to, bring a pencil and calculator. I'll have scrap paper for you. Again, that's going to be very basic math, so you probably won't need a calculator, but if you do, you cannot use your phone. What else to tell you? Might as well get acquainted with this now. I don't think I mentioned this before. On the front of these desks are chargers. So when you're sitting here doing your exam, you can plug your computers in right at the front. Also, for the exam, you guys will be facing the other direction because I'll be here and I just want to be able to see your screens because obviously the point of us being here taking it together is I'm making sure you're not looking things up on ChatGPT or opening your online textbook, things like that. It's a closed book exam. Yeah, so that's it. Also, again, one last time, whenever we do it, you'll have 50 minutes to take it. 50 minutes to answer about 50 multiple choice questions. If you're bad at testing or you just want more time, or you just don't feel like coming in, again, contact me. If we can make arrangements for maybe you to meet me in the office or us to meet online, if I can proctor your exam and make sure you're taking a closed book exam, I don't care how long it takes you. Take you 2 hours, I don't care. As long as I know you're not looking things up, it's all right with me. So any questions about anything from here up? All right, in that case, let's get started. It is 8:01. Really quickly, I'm gonna do attendance before I give you the attendance word. So Ashley is here, Barrett is here, Wooten, is Wooten here? Wooten is not here. Trahan, I think, is online. If you're online, you don't need to worry about it because you're gonna send me the email, so it's okay. I mean, send me the email, worry about it that way, but I'm saying when I'm calling names for attendance, don't worry about it if you're online. Hannah Diggins is not here. Clark is not here. Inca is here. Isaiah is not here. Let's see. Copes is not here. Smith, right? Is that Smith back there? Yep, Smith is here. Watson's not here, I think. Kaylee, not here. Lucas is here. He's here, right? Yep, Schaefer is here. Noah is not here. Cunningham is not here. Bartoli is not here. Is it Edwards? Nope, Edwards is not here. Wilkes. Yeah, Wilkes is here. Anybody I didn't call that's here in person? All right, well, we'll get started. First word for attendance is not a word at all. It is a name. And this is the name. It's this person's birthday. William P. Murphy. Later on, please don't do it now when I'm giving you a lecture, but later on, if you want to look that name up for extra credit and find out who that was, feel free. Send me a separate email. Don't send it in the same attendance email. Say, hey, I looked it up, and according to this source, William P. Murphy was the inventor of the Murphy bed. That's not true. I made that up, but you can look it up. Anyway, hopefully you guys are caught up, which is a good bring this up too. You're always going to want to watch a previous discussion, or excuse me, a previous lecture before you watch a current one. Especially now, given this weather, I'm being pretty flexible with attendance. So, for example, if you're here right now, but you haven't seen Wednesday's lecture, it's not going to be as beneficial for you, right? So, also on Monday, come Monday, as Jimmy Buffett would say, if you haven't watched the previous lectures, I would recommend just don't come in. Make sure you're caught up with the previous lectures before you watch the new ones, because they build on each other. A lot of times, it's cumulative. So anyway, that being said, this is where we left off. We were talking about proteins and protein structures. I really went into a long discussion the first time I recorded my lecture on Wednesday, which turned out not to record. So I don't remember exactly what the other video, I shared a different video with you. But I wanted to point out something that I don't think I said in that video. So I'll make sure you understand. For the exam, you're really going to be concerned with primary structure. Because primary structure is just basically describing which amino acid is attached to which amino acid. That's all you're concerned with. This one is attached to this one, is attached to that one. That's the primary structure. Then, as far as we're concerned, the rest of those kind of match together. This is a 100 level non-biology class, so it's not very important that you understand the difference between secondary and tertiary and quaternary structure. The point is, when we're talking about primary, we're just considering which one is attached to which one. And then, as I mentioned previously in the lecture, that dictates, right? Whichever one is attached to the other one dictates the form that it finally does take. And that's what this is all about, the form that it finally does take. So it is important for biology, but as far as the exam is concerned, again, it's not important that you understand the difference between secondary, tertiary, and quaternary. Maybe quaternary, I will point this out. So again, when we're talking about, from here up, we're talking about the single polypeptide, right? So a polypeptide, again, is a string of amino acids. It folds and it makes its final shape, right? That's what this is. But then sometimes a protein is more than one polypeptide. So when you have more than one polypeptide come together, we call that a quaternary structure. And it's probably, again, I'm probably not going to ask you that, but it is good to know to keep in the back of your head. So for example, not the next exam, but the exam after that, when we talk about cellular reproduction, where we talk about how there's an enzyme that attaches to your DNA, and it reads the code and it makes another copy of your DNA. And that enzyme, as we describe it in this textbook and in this course, we just say it's a protein. But in all actuality, it's a team of proteins that have come together, which would be quaternary structure. So any questions about that? All right, so again, here we have an example. On the left-hand side is the primary structure. I know it's kind of hard for you to see, but we have a lysine attached to a valine, attached to a phenylalanine, attached to a glycine, so on and so forth. You don't need to know what those words mean. Just know, remember, there's 20 different amino acids, and I was just saying the names of some of them. So they're all attached together. The only reason it even has this shape in this picture is so they can fit it all in one picture, right? That shape is useless. That's just the primary structure. But due to its primary structure and all the interactions between those R groups on these R groups on each amino acid, right? They all interact differently. Therefore, they have a different shape. And this is the final shape. And you would never need to recognize that, but it's just an example. And again, it's a good time to remind you, when it comes to proteins, their structure, I mean, a lot of stuff is really based on the function. Their function is really based on the structure. But this is even more so with the protein, right? Protein, it's all about the shape. Shape is very important for the protein, especially later in the semester when we start talking about specific types of proteins, which I've already hinted at. But mostly the proteins we're going to talk about are going to be enzymes, right? And enzymes are very, very dictated. Their function is very much dictated by the shape, more so than most other proteins. Which brings me to this point. If there's a slight change in the sequence, so again, we're talking about primary structure where this amino acid is attached to that amino acid, so on and so forth. If you change that even slightly, it could have a big effect. And that's also going to be very important later in the semester when we talk about things like mutations, because mutations cause this. And then after we talk about mutations, we're going to talk about stuff like evolution. And you can't even have evolution without this. Because what happens is something goes wrong, and we'll talk about this later, but something goes wrong in your cell. So when you make new DNA, it does it incorrectly. And therefore, sometimes you have a change in the sequence, because we haven't mentioned this officially yet, but it's the sequence in the DNA that dictates which amino acid is attached to which amino acid. So again, this is slightly important for this exam, but this one slide is actually quite important later in the semester because these concepts will come back. One example is this, hemoglobin. I'm gonna put it next to this just to make sure you're very aware that it's just an example. I'm not gonna ask any questions about this, but this example will come back later in the semester. Again, when we talk about mutations and evolution. But anyway, if you substitute a single amino acid for the polypeptide that makes up hemoglobin, it causes a disease. And again, we'll talk about it later. The name of the disease is sickle cell disease. I'll show you this. Here's what it looks like. Here we have the primary structure of a regular hemoglobin protein. Here we have the primary structure of one that's been mutated. You can see, once again, I'll remind you, the only reason it has that shape is so they can fit it all on one screen. It could be any shape because we're talking about the primary structure. But anyway, all we have is this. So 1, 2, 3, 4, 5, 6. We have a cysteine here. 2, 3, 4, 5, 6. Well, maybe they drew that wrong. Looks like the same one. Anyway, the point is, with this mutation, when you have this whole long chain of amino acids, only one of them is different. And because of that one difference, we have a different shaped protein. And it is kind of hard to see right here. But then, because of that different shaped protein, now you have completely different shaped blood cells. And again, this disease, it's not like that's it, that's the disease. Like, oh, you've got unattractive blood cells, that look funny. No one's gonna care about that, right? The fact that you have these sickle cell shaped blood cells actually lead to other things that are, bad for you. But again, we'll talk about that later in the semester. This is just an example. So any questions about that example? Okay, so again, basically everything I just said is now going to be here in writing. One difference is enough to cause a protein to fold into a different shape. And again, when we're talking about proteins, especially enzymes, it's very much all about the shape, right? So if you alter the shape, you alter the function. And that can cause diseases. Matter of fact, I'm going to give you another example. This is from the old textbook, I think. We're going to talk about this particular disease later in the semester. I'm definitely not going to ask you any questions about it on this exam. I might ask it about it on a later exam. But anyway, sometimes these misfolded proteins are kind of contagious in that not only are they misfolded, but then once they come in contact with other proteins, that causes them to misfold, which is unique. it's different than what I've described before, because I described before is the protein had the wrong shape because it had the wrong amino acid. And it had the wrong amino acid because there was a mutation in the DNA, right? So it was a whole process that got there. Here, it's just a severely misshaped protein that can even just physically misshape another protein by touching it. So there's the example of that. Again, you're not going to need to know this for the first exam. You might need to know it later in the semester. But these are called prions. And you can see here we have these normal proteins, kind of look like a pill. We've got these other ones that kind of look like a heart, these misshaped proteins. And when they attach to them, they just all clump together and they all get misfolded. And eventually, basically, it puts holes in your brain. And that's where that name, bovine spongiform encephalopathy. Basically, we're talking about encephalopathy, spongiform. It's basically turned your brain into a sponge, right? And you may have heard of that. Of course, you guys are a little bit too young. Maybe you haven't. There's something called mad cow disease. It was kind of big when I was a kid or a teenager. So yeah, you might not have heard it. Anyway, that was pretty big back then. There's Kuru. So these are all the same diseases, just for different animals. So Kuru is for humans. If you want to for extra credit, you can look into the history of that. That's interesting, because that spread in a very interesting way. And then if you hunt, if you're from around here, you might be familiar with this one. Fatal weight loss in deer, elk, and moose. But again, these are just examples. There'll be no questions about this on the exam. Do you have any questions? Okay, the next word for attendance will be cow. Let's have a cow there. All right, let's move forward. So again, I'm really hammering this point home because it's very important. When we're talking about proteins, it's all about the shape. Shape is very important to its structure, to its function, excuse me. But that being said, it's also important to note that its shape is sensitive to its environment. And this will be important here in a couple of weeks when we do the photosynthesis lab, photosynthesis/respiration. Because even though we don't talk about it a lot, we will mention it. Every step in the process of photosynthesis and every step in the process of respiration has a specific enzyme. A specific enzyme for this step, a specific enzyme for that step, right? So without the enzymes, those steps wouldn't happen, and those whole processes of photosynthesis and respiration wouldn't happen. But again, they're sensitive to the environment. So keep that in mind, because when we do those labs, this is the kind of stuff you're going to need to think about. So yeah, you're going to have your data that says, all right, when I put this much light, I had this much photosynthesis. When I use this color light, I had this much photosynthesis. That's the basic stuff. But then you need to think deeper and think about the environment that you're putting it in, because again, the environment will change its shape, which will change its structure. So let's talk about some examples. A change in temperature. You already know from when we first mentioned the word temperature, that when we talk about temperature, changing temperatures, what we're really talking about at the molecular level is molecules moving faster or slower, right? When we say things are heating up, that means there's more kinetic energy, right? Those molecules are moving faster. When we say things are cooling down, that means there's less molecules, there's less movement of molecules. But that's important, because what I didn't say, I don't think, is that basically, again, when things are heating up, you're putting energy into that system. Now here in this picture, we have some covalent bonds that's keeping this ribbon of a protein together, right? That's covalently bonded. But what's caused it to squish together like that in that particular shape are these dotted lines. And I hope you remember, because you need to for the exam, those dotted lines are hydrogen bonds. Of course, it's written right there. And hopefully you remember, because you need to know for the exam, that covalent bonds are stronger than hydrogen bonds. Covalent are actually the strongest, hydrogen are the weakest. So here we have these weak bonds keeping this thing together in that shape. So again, if you add temperature, if you add heat to it, you're adding energy to that system. And eventually what can happen is if it gets enough energy, it can just break those hydrogen bonds, because they're easy to break, right? They're the weakest of the bonds. So again, too much energy, what's going to break the bonds, it's going to ravel it. The word for that is denature. And then once it loses its shape, it can't lose its function. pH, that's another good example. So remember, what's causing these hydrogen bonds is that this oxygen has a slight negative end, slight negative, right? This hydrogen has a slight positive end. So they're slightly attracted to each other. You need to know that for the exam. So this one slide is a great review of stuff you need to know for the exam, even though I'm giving you new information. So we know that, right? Another thing you should know for the exam is that we talk about something being acidic, which is where the pH has gone down. right? What we're saying is there's a bunch of H plus ions, right? There's a high concentration of H plus ions if something is more acidic. So imagine that. This hydrogen is fully positive. It's not slightly positive. That's a fully positive. It's a proton, really. So that thing is going to be attracted to this oxygen. Or more importantly, I guess you could say that oxygen is going to be more attracted to that hydrogen. Right? So this hydrogen is fully positive. This one's only slightly positive. So that's quickly going to break those bonds so that that oxygen can attach to those, those hydrogen ions. Right? So again, lowering the pH is going to cause that to unravel because it's going to disrupt those hydrogen bonds. Meanwhile, increasing the pH, again, a reminder because you need to know this for the exam, means there's a higher concentration of these OH ions, right? OH minus, fully negative. So once again, if it's too basic, this hydrogen right here is going to be more attracted to that OH ion because it is fully negative. This thing's only slightly negative. So once again, if the pH is too high or too low, you can see now how that would cause it to basically unravel, right, and lose its shape, denature. And if it does that, it doesn't, it can't function. So yeah, again, for the exam, that's a little bit less important, but I do want you to keep that in the back of your mind once we start talking about other things, because almost everything we talk about until we get out of cells is going to be dictated by proteins, these enzymes that do these different jobs that we're talking about. And they're very sensitive. And then the last thing I'll mention is they're also, because of natural selection, I don't even know if your book talks about this or evolution, organisms are evolved to be in their environment. So for example, there's some bacteria that live in the hot springs of the Yellowstone National Park. Hot springs, right, in boiling water. But they are evolved for that, right? So when you put them in boiling water, they're like have stronger bonds. Whatever it is that's keeping their shape together does not dissolve, does not denature in that heat. Obviously, some things live in colder environments. Some things live in a more acidic environment. Think about all the bacteria that live in your stomach with all that stomach acid, right? So everything's evolved. But yeah, proteins are very sensitive to their environment. Any questions about that? Whew. All right, we might finish this chapter today. So again, big picture, let's take a big step back. Chapter 2 is all about chemistry. It was broken down into 3 main portions. We've been working on this last third main portion for a while now. That third main portion is all about biological molecules. There's 4 different categories. We've already talked about carbohydrates. We already talked about lipids. We just finished talking about proteins, so now we're going to talk about the 4th category, which is nucleic acids. I will say I'm going to go through this kind of quickly, because there will be a few questions about this on the exam, but I don't really hammer on it too much or ask that many questions, because later in the semester, we're going to come back and talk about nucleic acids again in a much greater detail. So this is more like an introduction, in my opinion. So some of this stuff will come back later in the semester. But anyway, nucleic acids, what do they do? That's one thing you might need to know for the exam. You'll definitely need to know later in the semester. They store information and they provide instructions for building proteins. That's basically it. You could look it up and probably get different answers, more specific answers, but those more specific answers would probably fall under the category or under the umbrella of one of these 2 things. And so if they talk about, DNA gets passed along from from a parent to an offspring, and that's the basis of heredity, well, that falls under storing information. And like I said previously, when we were talking about proteins and the primary structure and how this amino acid is attached to that one, which is attached to this one, so on and so forth. Again, it's the DNA and then the RNA, it's the nucleic acids that provide those instructions that say, attach first this amino acid, now that one, so on and so forth. There's 2 types. You definitely need to know this for the exam because this is the most basic information. There's DNA and RNA. You'll really later in the semester need to know the difference between them. Now it's just an introduction, but yeah, DNA and RNA. DNA stands for deoxyribonucleic acid. We'll explain that later. RNA just stands for ribonucleic acid. You can tell even by the names, they must be similar, and they are. They have very similar structures, so as you might imagine, very similar functions. Slightly different structures, so as you might imagine, slightly different functions. So any questions about this slide before we move forward and talk about some more about nucleic acids? Okay, Here's another term that you're going to talk about, that you're going to learn later in the semester. So this is just an introduction. It might not be on the first exam, but there's going to be an exam that covers this a lot later in the semester. A gene. I'm sure you've heard this term before. A gene is a unit of inheritance encoded in a specific stretch of DNA, and it programs the amino acid sequence of polypeptides. So what I was saying earlier about how nucleic acids dictate the sequence of amino acids, this is what I'm saying, right? I'm just giving you a name for it now. It's a gene. So when you're talking about DNA or nucleic acids, when you're looking at DNA, for example, portions of it, this is what they do. They provide instructions on how to build a polypeptide. The instructions are written in a chemical code that has to be translated. That is something we're definitely going to cover later, and I'll ask you about later. But for now, this is just an introduction, so I'm going to put it next to this. Definitely not going to ask you this on the first exam. But yeah, it's a code. This code had to be translated because we have this nucleic acid language, which, as you're going to learn later, is made up of 4 letters. So you know, like how our language has 26 letters, this language has 4 letters. So we take that and then we translate it to a different language, which is the protein language. And the protein language, in a sense, has 20 different letters because the protein language is just saying, attach this amino acid to that amino acid, right? It just dictates which of the 20 amino acid is next on the chain of amino acids. So again, you really can't think of these as languages. Our language, English language, has 26 letters. The nucleic acid language has 4 letters, and the protein language has 20 letters. Not that that'll ever be a question, but it will help to know that, to help understand some stuff. So any questions about what's written on this slide before I show you a picture of what's on this slide? All right, so here we have a long, well, what we would have is a really long strand of DNA, but we've zoomed into it, to this one particular portion of it. And in this particular portion from here to here is a gene, right? That gene, and we're going to talk about this later in the semester, that code is then copied down because we can't use it directly there. So we copy it down like you would, like that's a book. And you want to write down instructions, there's a piece of paper, right? That's basically what's happening. You've got this whole book of instructions, but you only need that one set of instructions. So you open the page to get to the set you need. You write it down. Now you've got just the one piece of paper, the instructions you need. You take it to the machine that translates it. It reads it, says, all right, first I need this amino acid. Okay. Now I need this amino acid. All right. Now I need this amino acid, so on and so forth. So again, we had to translate this nucleic acid language to the protein language. And again, you're going to learn about that later in the semester. So any questions so far? All right. Nucleic acids are the key macromolecule in the continuity of life. Because again, it's the instructions on how to build all these proteins. And now those instructions get passed on from parent to offspring. So that's what we mean by the continuity of life. Without that, life would just have to start over every time something died. And, that wouldn't happen for the most part. So that's important to know. Not necessarily for the exam, but it's important to keep in mind. And also, again, I haven't really hammered on this enough. I've mentioned it a little bit. But like I said, if you take away all the water from the cell, by weight, over half of it, what's left is going to be proteins. So that should tell you how important they are. And they are because enzymes are proteins, and enzymes are the things that make all these chemical reactions in our cells happen. Without those chemical reactions, we would not be alive. So again, very important. So again, this is sort of like information I've already said before, and I'm just saying it in a different way. And again, there's 2 different types, DNA and RNA. Any questions? All right. Well, let's talk a little bit about the differences between the DNA and the RNA. You might need to know that for this exam. You'll definitely need to know it later. DNA itself is the genetic material. So when we say, the continuity of life, and it goes from parent to offspring, that's what's happening, right? It's the DNA that's getting passed from the parent to the offspring. It's found in all living organisms. So anything alive has DNA in it. Even some non-living things have DNA in it. You'll learn about that later too. Viruses are not technically alive. We'll talk about that why later. But they have DNA. Some of them do. Some of them have RNA. That's a different discussion for later. But yeah, that's DNA. Any questions about DNA? Again, it's the genetic material. And the next one, again, RNA. This is mostly used for protein synthesis. What's that star for? Oh yeah, just like everything else. You're gonna learn about that later. So yeah, when I say protein synthesis, this is something that you're going to learn about later. Right now, I'm just introducing you to it. But yeah, that's what RNA is. RNA is mostly used for protein synthesis. It has other jobs as well that we'll touch on. But even then, we don't even get into the other stuff. When we talk about RNA later, it's almost always going to be talking about how different types of RNA come together to make protein. But yeah, and again, like I said, DNA uses RNA to communicate. Like I was saying earlier, when we showed that picture of the gene, I said we have this whole DNA, this whole chromosome with all these different genes on it. We only need instructions on how to build one protein. So we're just going to copy it down, take that one page, and use that to make the protein. That's what the RNA is, right? RNA is that copying. Like, I'm just copying down this one gene. So I don't need all the instructions, I just need the one gene. That's what the RNA is. Any questions so far? Don't hesitate to tell me to slow down or speed up for that matter. Okay, let's see. So once again, let's take a big step back. For this exam, when we're talking about these 4 different biological molecules, you need to be able to compare and contrast before, right? Identify them in different ways. Realize how they're the same, realize how they're different. One of the ways I said we're going to do that, the common theme would be the monomers and the polymers, right? So when we talked about carbohydrates, the monomers were the monosaccharides and the polymers were the polysaccharides. When we talked about proteins, what we just finished, the monomer is the amino acid and the polymer is the polypeptide. When we talk about lipids, that wasn't technically monomers and polymers because they weren't repeating parts, but you did, for example, need to know that a triglyceride, which is a dietary fat, is a glycerol molecule with 3 fatty acids. You needed to know that the phospholipid, which is in our cell membrane, that's a glycerol molecule with 2 fatty acids. And when we talk about steroids, you do need to know that that's 4 carbon rings fused together. So that being said, let's talk about the monomer and the polymer of nucleic acids. Whether you're talking about DNA or RNA, it's the same. The monomers are called nucleotides, and the polymers are called polynucleotides. Nice and easy. Sort of like the carbohydrates. It's nice and easy, right? Monosaccharide, polysaccharide. With this, nucleotides, polynucleotides. I guess it'd be a little bit easier if they called them mononucleotides, but they don't. Anyway, much like when we were talking about proteins, we said, all right, the monomer of a protein is an amino acid, and you needed to know the parts, right? So you have that one central carbon attached to the hydrogen, a carboxyl group, an amino group, and then the R group, which was the one that R group, that's what dictated which of the 20 amino acids it is. But just like that, you also need to know this. When we're talking about the monomers of nucleotides, you need to know the 3 parts. There's a 5-carbon sugar, there's a phosphate group, there's a nitrogen-containing base. You need to know that. And for shorthand, I bolded a few words. If you can just memorize, like if you do flashcards, if you just memorize sugar, phosphate, base, you should be fine. Sugar, phosphate, base. Yes, the sugar is a 5-carbon sugar. It's a pentose. That doesn't really matter for anything in our discussions this semester. Yeah, it's a phosphate group, not just a phosphate, but for this level of learning, that's okay too. And again, yes, the base right over here is nitrogen containing, but as far as we're concerned, that doesn't matter either. So any questions about that? Before we move forward, I'll point this out. So again, when we were talking about amino acids, so again, proteins, not nucleic acids, but when we're talking about proteins, the monomer of the proteins, which is the amino acid, and that central carbon that was bonded to those 4 different parts. Remember, again, I said it was that R group that dictated which one it was, or defined which one it was, right? There's 20 different amino acids, and all 3 parts were the same. The part that wasn't the same was that 4th part, which was the R group. That's what says this is this amino acid instead of that one. In this case, it's the base. So we're talking about nucleic acids. The sugar and the phosphate, always the same. The only thing that's different is the nitrogen, the base, right? That's what dictates which of the 4 it is. So remember, with amino acid, there's 20 different versions. With nucleotides, there's 4. Technically 5, we'll talk about that later. But again, it's this that varies, right? The phosphate group's always the same, sugar's always the same. It's this that's going to say, all right, this is this particular nucleotide instead of the other. So any questions about that before I show you a different picture of a nucleotide? All right, here's the thymine. This is just an example. You don't need to memorize this. I'm not going to ask you what thymine is. I'm not going to give you a picture of this and say which base is this. It is thymine, but I'm not going to ask you to memorize that. I'm not going to give you a picture and say, all right, which part's the phosphate, which part's the sugar, which part's the base. I'm not going to do any of that. So this is just an example. But yeah, this is one of the four. This is one of the four DNA bases. There's either an A, a G, a C, or a T. This one's a T. All DNA bases have the same phosphate group and the same sugar. The only thing that's different is this base. This happens to be a T, which is unique from the other three. And this over here, it's the same thing. This is a more simplified drawing. You still have the sugar, you still have the phosphate, you still have the base. So anyway, any questions about that example? Okay. Next word for attendance is going to be blue, like the color of that sugar. Blue, blue, blue. All right. So as I've already mentioned, when we're talking about DNA or RNA, there's monomers, and these monomers, these nucleotides, there's four versions of them, right? So for DNA, the four versions are this, and you might need to know this for the exam. You'll definitely need to know it later in the semester. If you need to know it for this exam, it's only going to be the letters. You do not need to know the whole name. But the four bases for DNA are A, G, T, and C. Not in that order either, doesn't matter. The order does not matter. A, G, T, C, C, T, G, A, doesn't matter. Those are the four nitrogenous bases. On the next slide, I'll show you some pictures of what they look like. Not that it matters too much, especially at this point in the semester. Later, their shape and their size will be slightly important, but for now, not important at all. So again, those are the four bases. Here they are in drawings, right? We have A, G, T, and C. Now for the exam, this will never be important, but for understanding stuff later in the semester, this will be important. Notice that A is a double ring and G is a double ring. T is a single ring. C is a single ring as well. That will be important later. Not for the exam, but to truly understand how things come together, that fact will be important. Yeah. So anyway, any questions about this slide? All right. So let me ask you this. We're talking about all these different individual monomers here. And of course, here we have a picture of them all together. But what kind of reaction is going to bring these monomers together? This is a good review because you need to know this for the exam. And it's been a common theme throughout the semester, or this portion of the chapter when we're talking about monomers and polymers. What kind of reaction is it that brings these monomers together? And I give you a hint. In the products, is water. The reaction produces water. What kind of reaction is that? Again, you'll need to know it for the exam. Anyone wants to guess? Don't be shy. Nothing wrong with getting it wrong, because then I'll know what you're thinking. It's better for me to know, like if you have a wrong answer, I'd like to know it so I can take that and help you understand why it's wrong and give you some hints of why, how to get to the right answer. But if no one wants to say, again, the answer is a dehydration reaction. So again, if you produce water, of course you don't see the reaction like I have previously. I've shown you the reaction. But again, remember, if you're producing water, if there's water in the products, that's a dehydration reaction. Just like your body, when you're producing water, whether it be via sweat, or you're spitting a lot, or peeing, whatever it may be, right? You're producing water, therefore you are becoming dehydrated. Anyways, yeah, it's a dehydration reaction that brings these nucleotides together to make polynucleotides. Again, this is made by covalent bonds. Later in the semester, you'll need to know this. Right now, you definitely do not. So yes, maybe you need to know the covalent, but you definitely don't need to know what's from the sugar of one and the phosphate to the next. At least not now, maybe later. So again, hopefully you were thinking about that, because I did just say that these nucleotides have 3 parts. So hopefully you were wondering, all right, if they have 3 parts, which part, when we bring them together, which part is attached to which part, and now you know. Sugar of one to the phosphate of another. The base does its own thing. We'll talk about that later. So the only reason that's even helpful for you guys, the only reason I bring it up is because there's a term you will need to know later in the semester. It's called the sugar phosphate backbone. So when we're talking about a long strand of DNA or even RNA, Talk about that sugar phosphate backbone. Again, later in the semester, that'll be important. Now I'm just introducing you to it. So, any questions about this slide? Oh yeah, this is not even from your textbook. This is from the old textbook. The old textbook went into pretty deep detail about this in the first chapter, like in the beginning of the semester, and then also went later in the semester. We got into more detail, so I kept it. But again, for this exam, exam one, this will be very unimportant. Later, you will need to know this much detail. All right, so now we're going to talk about the differences between RNA and DNA. Maybe you'll need to know that for this exam. You'll definitely need to know it later in the semester. Does anybody know any of the differences? No? All right, I'll give it to you. Here we go. DNA is double-stranded, and RNA is not, right? Well, actually, we won't even get there yet. I'm going to have it all on one side. I guess we'll talk about DNA first. Again, this is not your textbook. I'm going to go pretty quickly because I'm not going to go through this much detail in your exam. But yeah, DNA is a double-stranded molecule. This will make a lot more sense on the next slide when I show you a picture of it. What I mean by that is we have these polynucleotides that are coiled around each other. So DNA, I'm going to try to draw this, probably won't work. And again, the next picture would be better. DNA is not just one polynucleotide. DNA is two polynucleotides. Of course, here I've just drawn them parallel to each other, or tried to. But as you can see in this description here, they're not parallel to each other. They actually coil around each other. And that's what we call a double helix. That would be more relevant in the next slide. So again, like I already mentioned, we have a sugar phosphate backbone. So this little red line right here for this one polypeptide, that's because there's a sugar covalently bonding to a phosphate, covalently bonding to a sugar, so on and so forth. So what's keeping these two strands together is this, the second bullet point. It's the bases, which I haven't drawn, right? So we've got these bases sticking out. That looks so bad. But anyway, we have these bases sticking out. And those things, and I haven't drawn this yet either, I'm not going to draw all of it. These are hydrogen bonded to each other. I'm trying to draw little dots there, right? Each of these bases is hydrogen bonded to the other. So that's what's keeping those two strands together, as these hydrogen bonds. And again, for this exam, that's not very important, but later, that will be important. You're going to need to know all these details. So I'm glad I can put it in your brain now so it can marinate between now and then. But yeah, that's what keeps them together, is those hydrogen bonds. There's certain rules for this, so they don't just pair up willy-nilly. It's always A's that go with T's and C's that go with G's. You might need to know that for this exam. Definitely going to need to know it for another exam later in the semester. A's always go with T, C's always go with G. Again, all this information is going to be on the illustrated in the next slide, so hopefully make more sense. All right, any questions before we see the picture of it? All right, so here we go. Again, when we're looking at this DNA, it's not a strand of polynucleotides, it's two strands of polynucleotides. They're hydrogen bonded to each other, and then because of their chemistry that we don't even get into, there's things that cause it to twist. But even for us, that's not very important. The most important thing for us, anyway, is that we have two strands of polynucleotides, and that they're held together by these weak hydrogen bonds between the bases. And the bases are always C going with G and T going with A. The shape for us does not matter. They could be drawn realistically like that, or it could be drawn in my horrible way where it's two parallel strands. For us, it doesn't matter. If you're really looking into this and really looking at the physics and chemistry of DNA, which we don't get that deep, then the shape does matter. But for us, it doesn't really matter. Again, two strands of polynucleotides, each one strand is held together by strong covalent bonds. And these two individual strands are held together with each other via hydrogen bonds between the bases. Very important stuff later in the semester. For now, just an introduction. So, any questions about that? All right, we've got 10 minutes. I feel like we can finish this off. So like I said, a little preemptively, there are some differences between DNA and RNA. No one answered, so I'm going to go forward with it anyway. Let's talk about how they're similar. Again, like we've already mentioned, they are polymers of nucleotides. We already said that. They are polymers, which are called polynucleotides in this case. The monomers are called mononucleotides, or excuse me, I messed my setup up because I said it earlier. The monomers are called nucleotides. So that's the same. We already mentioned this, the three parts of the monomers, sugar, phosphate, and base. right? So technically a 5-carbon sugar, and technically a phosphate group, and technically a nitrogenous base, but whatever, sugar phosphate base. That's how they're the same. Hopefully, looking at that picture, you can see some of the differences, but let's talk about them. The 3 differences. RNA has a different sugar, hence the different name. Its sugar is called ribose instead of deoxyribose. That's really unimportant. I mean, that might be a question later in the semester. But to understand how DNA and RNA work, that's trivial. This next one is important to understand how this whole process works later in the semester. Instead of a T, RNA has a U. So RNA, just like DNA, has only 4 bases. Remember, DNA has A, C, T, and G. Now, RNA doesn't have A, T, C, and G. It has A, U, C, and G, right? Because it doesn't have a T, it has a U. And that will be important later in the semester to understand how DNA and RNA work. Like that picture I showed you where we had the DNA gene, and then we had that little strip of RNA, and then we had the protein. You're going to be asked to do that. I'm going to give you a code of DNA, and then you're going to need to write that code in its RNA version, which is going to require you to know that it's a U instead of a T, and it's going to require you to know A's and T's go together and C's and G's go together. Anyway, then you're going to take that code, and you're going to figure out which amino acid comes next, so on and so forth. So this will be important later in the semester. And then again, the other one, a little bit less important, RNA is single-stranded, and DNA, as you already know, because we just talked about it, is double-stranded. That will be a lot less important. So again, to understand how DNA and RNA work, these 2 bullet points don't matter much in this 100 level class. But that third one, or that middle one, that does matter. Also, as you're going to learn later, when we say RNA is single-stranded, that is true, but it can get a little tricky. Because a lot of times those single strands kind of flop back up on each other. And if you're just looking at that one portion, it looks a lot like DNA because you have a strand of polynucleotides, hydrogen bonded to another strand of polynucleotides. The only thing that makes it single-stranded, even though they're attached to each other, is it is just that one strand. Instead of 2 strands coming together, it's one strand that folded up back on each other. So any questions about this? The next slide is just a picture of U, because like I said, RNA has a U instead of a T. So the old textbook included, again, this is not your textbook, the old textbook included a picture of it. There it is. There's a U instead of a T. Not that it matters. And I bet, unless you guys are really, really observant, you probably can't even notice a difference between that and the other pictures I showed you. Maybe you're like, oh yeah, look at that, that's single. This is 1 ring, and some of the other ones he showed me were 2 rings. And that's okay, because at this level, 100 level, you don't even need to know the difference between the U's and T's and C's and G's and A's. You just need to know that they are different. But anyway, also, you might notice if you're really observant that this color is red instead of blue. And again, that's just demonstrating that it's a different sugar. It's ribose instead of deoxyribose. And again, for us, that is completely irrelevant. It does not matter. But as you can imagine, in deeper biology, it does matter. But for us, it's trivial. All right. So the last thing I'll say, I just remember what I was mentioning about lab. You can help. Come to me for help. I'm here to help. And one of the questions I got from one of you, which is a good question, I want to say it to everybody before we break up here. So this week's pre-lab, the one that's due today, is for the, diffusion and osmosis, is that right? Yeah, diffusion and osmosis. So you're going to watch a video and answer some questions. You're going to read an introduction. You're going to answer some questions. And then afterwards, you have some hypothesis questions to answer. And they're all highlighted in green. Because the question was, hey, where does the pre-lab end? It's a good question because it's not as clear as they are in the other ones. So yeah, just look for the hypothesis questions. They're highlighted in green. The last word for attendance, other than, remember, don't forget to send me a question. Of course, you guys in person don't have to do any of that. Just send me an email. But if you're watching online, also don't forget a question. But the last word for attendance is sugar. And that's it. Hopefully, weather permitting, I will see everybody on Monday. You guys have a good weekend. And remember, please, I'm here to help you. You do not have to do any of this alone.